All right, so as Jen said, I'm Eric Sokol, and I work at NEON here in Boulder. And today I'll be presenting some work that the LTER Metacommunity Synthesis group has been doing. Specifically, I'll be talking about using long-term data to understand links between environmental variability and metacommunity stability. So I noticed that one of the emails that went out said we'd be talking about disturbance, and that is one of the focuses of the working group, but it's not something that we have results that we can present on yet. So actually, I'll probably be soliciting for some input from you all at the end of the talk. Okay, and so as I said, this is really the work of a group of people, pretty large group of people. We have probably about 25 or 30 folks that have contributed to the working group, and I have listed here some of the core contributors. So I want to specifically point out Nathan Wisnowski and Chris Swan are my co-PIs on this working group. And then also Tomas Laney has led a lot of the work that I'm going to be presenting today, so the development of a community turnover metric that we're using. And so one of the main motivations of this working group is that many investigators across the LTER focus on, well, when they ask questions about biodiversity, we're trying to understand biodiversity at multiple scales. And a lot of field experiments or field observational studies look something like this. So this slide here is a picture of ponds that were built at the Kellogg Biological Station pre-LTER. I like this image because it really emphasizes sort of how, we sample discrete units in space and ecological landscapes often. But what we do is we try to understand both what's happening locally, but also in aggregate amongst all our observational units. And there's some assumptions and kind of some sort of relationships we have to draw to expand our study system to the natural landscape. Now, also, I wanted to give a shout out to KBS because, we're the Metacommunity Working Group, and this is where a lot of Metacommunity work originated. However, they didn't do that work in these ponds. I believe they used cattle tanks that they set up after these ponds stopped being used in the studies. So generally, we think about diversity like this in modern ecology. And this concept actually is half a century old now, but the idea of dividing biodiversity up into alpha, beta, and gamma diversity. So alpha diversity being measures of local diversity, so either richness or evenness at a local scale, beta diversity as a scaling metric, and then gamma diversity describing the regional diversity. So thinking about ponds, since they're nice discrete units in the landscape, we can measure alpha diversity in each observational unit. Gamma diversity is a way of estimating our regional species pool. And then beta diversity is some measure of variation in community composition and turnover through space. And there's a large number of ways to calculate beta diversity, lots of different ways to conceptualize beta diversity. One way is the multiplicative relationship. So you divide your regional diversity by your mean alpha diversity, and that gives you some measurement of scaling. How does each local community contribute to the regional species pool? So to understand or interpret biodiversity, we have to apply all those concepts in one overarching framework. And so we need to account for scaling. And metacommunity ecology provides a framework to account for these relationships and scaling. And when we think of these relationships between alpha, beta, and gamma diversity, really that looks like a conceptual drawing of a metacommunity, right? Okay, so what is a metacommunity? So it's more than just a group of observational units in a landscape. So really, the metacommunity concept is thinking of connected communities and realizing the importance of local interactions and how they influence local community assembly dynamics. So this includes competition, factors affecting local demographics, and other things. But also it recognizes the importance of dispersal amongst the patches within a landscape. And there are a number of different hypotheses that take into effect the relative importance of dispersal amongst our units in the landscape, and then also the importance of that relative to local interactions. And so really different levels of dispersal, functional diversity, and habitat heterogeneity affect the community assembly dynamics that we observe in the landscape. And so Logue et al. In 2011, in a review of metacommunity ecology, did a really nice job organizing kind of the different sorts of community assembly hypotheses in the metacommunity framework. So we can have different levels of dispersal, different levels of habitat heterogeneity, and different levels of functional diversity or functional equivalence amongst the species in the metacommunity. And there are a number of metacommunity paradigms that Leibold and others described in their seminal paper that came out of their NC's working group. And they talked about neutral models, patch dynamics, mass effects, and species sorting. And these 4 different, what they call metacommunity paradigms, are really, I like to think of them as signposts or reference points within this dynamic space that give us an idea of what sorts of community assembly dynamics begin to occur given a set of parameters. So like if we have high equivalence amongst our species, we're more likely to observe neutral model dynamics. If we have high habitat heterogeneity and a lot of dispersal, we start to see mass effects. Okay, so what is the utility of the metacommunity framework? So, I mean, this can get pretty complicated trying to put all the different community assembly hypotheses into one framework. But on the other hand, having a framework sort of unifying different hypotheses, putting them in context with one another can be really useful. And so there's been broad interest across the LTER in applying metacommunity theory. So in 2015, we had a working group or a workshop at the All Scientists meeting where we essentially wanted to see how folks were using metacommunity ecology across LTER sites. And so there's a general consensus that folks apply the concept across LTERs. There's lots of good publications out there from different LTER sites. At the meeting, we had a good number of participants from across the LTER network identify as folks who are interested in applying the metacommunity concept. And there were a good number of LTER sites represented in that room. So there's broad interest in applying the concept across LTER. And we think that the utility of this is it gives us a way to put community ecology and questions about biodiversity in this macro scale context. So this figure is from Heffernan et al. In Frontiers in 2014, describing macro scale ecology and some of the concepts of why it's important to think across scale. And metacommunity ecology essentially gives us a tool and fits really nicely into thinking about how things scale up from local to regional. So we can't really understand what's happening at the regional scale with an entire metacommunity without thinking about how the different patches within a metacommunity interact. And then also it gives us a framework for thinking of these cross-scale interactions. So how do regional characteristics and species pool characteristics affect what's happening at a local scale? Okay, so when we're using the metacommunity framework, how do we interpret biodiversity? And is it appropriate to infer process from pattern? So this has been a debate in the literature for a while. And so first I'll give an example of a success at an LTER site where folks have been comparing, or in a comparative study to assess how dispersal affects turnover along an elevational gradient. So Mike Willig and Stephen Priestley published this paper, which is a really nice example of essentially hypothesis testing with a study where they're comparing metacommunity characteristics for different groups of species. So rodents obviously don't fly, bats and passerine birds do fly. And so they set up a series of predictions to test in terms of what does community turnover look like along an elevational gradient. So here in the figures on the right, the y-axis is elevation and corresponds with the forest type. The x-axis is essentially a species list. And so we can see for rodents, there's specialists in terms of which elevation they make a living. And for things that are flight capable, there's much more overlap in community composition amongst the forests. So there's a strong influence of dispersal when comparing these different groups of organisms. And so they were able to look at a pattern and infer the importance of dispersal as a process. However, a lot of times folks set out to conduct an observational study and see a pattern and try and infer a process from a pattern. But so for example, here we had a study where we looked at community composition in ponds. And it's tempting to infer an underlying process from the diversity you observe in a snapshot of, in this case, zooplankton in our ponds. But there's a problem with equifinality. So through modeling, we're able to show that multiple underlying processes could create the pattern that we observed in the landscape. And so this kind of has been the motivation, at least for me, for thinking about how do we look at metacommunity data when we have a temporal dimension. So this gives us a much better ability to think about what's happening with dynamics. So in observational studies, we can look at trends through time and have a better idea of the processes that are occurring. So our goals in this working group are to apply the metacommunity concepts to interpret biodiversity at multiple scales through time. And specifically, we're looking at how metacommunity characteristics determine how metacommunity stability scales. And so stability in the structure of the metacommunity is essentially an emergent property that we're interested in when we look at metacommunity data sets. And so a real simple graphical representation of our hypothesis and also a hypothesis that a number of studies have recently been addressing is essentially thinking about how dispersal relates to regional stability. So if we have this simple example of a metacommunity and we're looking at total abundance of community 1 and community 2, if they fluctuate out of sync or have compensatory fluctuation, then we see stability at the metacommunity scale. So that's pretty intuitive. And then alternatively, if the two communities are fluctuating and it's synchronous, we see amplified instability at the regional scale. And we expect this first condition to occur under low dispersal in a metacommunity, and the second condition to occur when there's high dispersal in a metacommunity. Okay, so I'll stop right here just to see if there are any questions, and then we'll continue if I don't have anything in the Q&A.
So just to remind you all, if you have any questions, look for the Q&A button down on the bottom of your screen. At least it's on the bottom of mine, and you can type your questions in there. Eric, it usually takes a minute or two for folks to kind of find it. Okay.
Yeah, and I don't have the chat window open. I'm trying to find where to open it. So just in case.
So we're monitoring the chat as well. So if folks have a burning question, can't find the Q&A button, you can type it in chat. And it will likely go to all participants though. Yeah, I think feel free to move on, Eric. And
Right.
actually, one question just came in, Eric. Now, can you see it?
Oh yeah, I do.
Okay, there you go. Maybe read it out loud.
Okay, so the question is, what defines a high or low dispersal metacommunity? And so there's a couple, that's a good question. There's a couple different ways to address dispersal with metacommunities. So I'll go back to my little diagram of metacommunities here. So high dispersal can refer, first of all, to essentially the magnitude of movement between patches. So thinking back to like the neutral model, that's that M parameter. So essentially, you know, a higher M, you have a higher proportion of organisms moving between patches. But there's another dimension to it too, which is what's the shape of the dispersal kernel. So does the probability of something successfully moving diminish with the distance that is trying to move away within the patch? And in terms of this like oversimplified hypothesis here, I'm kind of wrapping up both of those concepts into dispersal. So we would expect synchrony to increase both with an increase in the magnitude of dispersal, but also with an increase in the distance that organisms can disperse. I guess there's a third component to that, which is dispersal equivalent amongst the species within the metacommunity. And that we're not really addressing at this stage. And then also a small question, how do we define stability? And that's actually what I'm going to talk about next. So I'll move on to stability. Okay, so we're going to be, as I was saying, we're interested in metacommunity stability. So first I want to talk about how we define stability. And so here I have some figures from Donahue et al. In Ecology Letters, where they described the components of stability really well. And so if we think about total biomass at a site and how it fluctuates through time, we can think of first just how variable that is, kind of the background variability, and you know that's the variance. We can measure that by taking the variance through time or coefficient of variation, something like that. There's also these components that are often associated with a disturbance event or perturbation of some sort. So for example, we can have a disturbance where the biomass is decreased markedly and its recovery time is related to its resilience. So longer recovery time is lower resilience. Here, a short recovery time is high resilience. And then resistance is kind of how far away from the norm that the disturbance pushes our response variable. So these are the different components of stability. I'm going to be talking about or focusing on just this variance component right now. So like I said at the beginning of the talk, we're not going to be addressing discrete disturbance events at this point in time. We're mostly going to be talking about how to approach data sets where we're measuring just the overall variability in local and regional aspects of the metacommunity through time. And a common metric, as I was saying, for stability, well, a common metric for variability is measuring the coefficient of variation of this time series. And then a common metric for stability is the inverse of that, so 1 over the coefficient of variation, or something similar to that. So sometimes it's 1 over CV squared, or sometimes variability is measured with CV squared. Okay, so I've just been talking about a univariate response metric, like total biomass. But Michele et al. In 1999 published a paper that's really influenced me and a bunch of my colleagues, where they described these two aspects of variability. So there's aggregate variability. So when we have a community, and in this case, it's multiple species within a community. Aggregate variability refers to the total variability of all species within the community. But then there's also compositional variability, which is through time, how does the composition of the community change through time? So they have these kind of 4, again, examples or reference points within this aggregate variability and compositional variability space. And so if both species remain static, the total compositional aggregate variability is relatively stable. But an important concept to point out is that we can have high variability in the aggregate metric or aggregate biomass, while community composition remains relatively steady. And conversely, we can have a relatively stable aggregate biomass while community composition is changing when we have compensatory dynamics. And in this framework, this idea of synchrony between the species really describes how the individual species level variability scales to the aggregate or to the community. And so I think it's a really important concept to think about. There's these aggregate dimensions and these compositional dimensions. And importantly, if we just look at aggregate metrics, we can miss what's going on with community composition, and vice versa. If we're only looking at composition, we can miss what's happening with the aggregate metric. Okay, so I've talked about community stability. So now we're going to go up one more scale in organization and talk about metacommunity stability. So there have been a number of papers, Long and Leroux put together a framework for thinking about metacommunity stability, where they described how we can look at community aggregate biomass and think about how that's related to the biomass fluctuation in individual species or their populations at the different sites that make up the communities, but also think about how fluctuations at the patch level for the communities scale up to affect total metacommunity stability. So in this case, we can think of productivity in the community, in communities 1 and 2, and how they fluctuate together to affect total metacommunity productivity. And this figure is from a paper by Wilcox et al., where they actually did a meta-analysis to look at these, to look at how metacommunity stability scales across a bunch of grassland or plant data sets. And so the take-home message here is that, interestingly, there's a similar multiplicative scaling that can be applied to stability that I talked about earlier in the talk. It's analogous to how biodiversity scales in a metacommunity. So remember, beta diversity is gamma diversity divided by your mean alpha diversity, if we're thinking about the multiplicative framework. Similarly, there's the synchrony metric that is basically the regional variability divided by your mean local variability. And so that's derived and described really well in these Wang and Leroux papers, and Wilcox et al. Do have this really good study applying this concept across data sets. And Wilcox et al. Showed basically that spatial asynchrony can stabilize aggregate properties of metacommunities. So, for example, in this case, we have high synchrony between different communities, and there's high variability at the metacommunity scale. And then in these other two cases, they have compensatory dynamics at the community level promote stability at the metacommunity scale, and also potentially asynchrony among species could potentially stabilize the regional, stabilize the metacommunity at the regional scale as well. Okay, so that's sort of the aggregate approach to that's sort of the aggregate metric approach to looking at metacommunity stability. So what we're working on in this working group is kind of adding in compositional stability. So the approach that I just talked about, say we have patch 1 and patch 2, and we have biomass that we're measuring of 2 different species, orange and blue through time. If those species are asynchronous at each patch, then that promotes stability at the patch scale. If those patches' total biomass are fluctuating in an asynchronous manner, that can promote stability at the regional scale or metacommunity scale in this aggregate biomass metric. So that's what we just talked about that previously, that Wilcox et al. Looked at, and that Wang and LaRoe derived that relationship. However, there's another approach that we can take, which is if we look at how species fluctuations affect essentially the regional dominance of each taxa and their representation in the species pool. And so essentially thinking about how fluctuations at the local scale affect the composition of the regional species pool. And this also can inform what's happening with the aggregate biomass metric at the regional scale. So essentially this part was previously missing from the framework, and we think that it's worthwhile and informative to explore. So essentially we're looking at the emergent property of composition of the regional species pool. And we think it'll be a useful contribution to thinking about how local scales to regional in terms of biodiversity and metacommunities. Let's see. So there's a couple of, let's see if I have a questions section now. I have a couple questions popping up, but I'll address them in a minute or two. Okay, so the goal of our meta-analysis is to address the question, does compositional stability scale differently across ecosystems? So here we've sort of adapted the Micheli et al. Figure to think about how compositional variability can scale in metacommunities. So essentially we're addressing the question, why look at scaling of composition? And the reason is because we think there could be situations in which local community compositions are changing differently through time that could promote stability in the regional species pool. So in this figure here, we have the inset panels are all, the y-axis is composition, whoops. So the y-axis, you can think of it as like scores for the principal component analysis of community composition. So this is like community type 1 up here and community type 2 down here. And as things fluctuate through time, the community composition is changing. And then this bolded line is the metacommunity composition, or the composition of the species pool. So we can have, similar to Micheli et al. At the smaller scale, we can have stasis, where both communities stay the same through time and the regional species pool stays the same. Or we can have fluctuations at the local scale, but it still maintains a relatively consistent species pool. And then there can be other situations. For example, if there's synchrony in the fluctuation in communities, it can create instability at the species pool scale for the entire metacommunity. So that's our motivation for looking at this compositional scaling metric. So back to our conceptual figure, we'll be focusing in on how to look at asynchrony in essentially the fluctuations of biomass or relative abundances of different species and how that affects community turnover in the species pool. Okay, so I figure at this point, folks probably have questions because I showed a couple of graphs that have a lot to digest. So I see, let's see, a question of how is synchrony approached, covariation. So the Wang and LaRoe papers in ecology letters describe how the synchrony metric is calculated. I'll give some examples later on that may kind of give you a better idea of what the number actually means. So I'll address this question in a little bit. Also, another question says, I would expect higher dispersal to homogenize the metacommunity, not lead to an unstable metacommunity. So that's a good point. The way some of the studies and various hypotheses address this issue is thinking about it in terms of if you have really high dispersal that's homogenizing, it essentially makes the entire meta-community act as one community. And so if you have a fluctuation in one direction or another, all of your patches kind of go in that direction. And so if you have, say, a species extinction, that species becomes extinct at every patch within your meta-community and then can't be rescued. So the alternative is if you have each of the patches fluctuating in different directions, if a species goes extinct in one patch, it's able to be rescued from another patch. So the idea is that if you have some dispersal but not a ton, that's the most stable scenario. If you have no dispersal at all, then it could potentially be unstable because you don't have rescue effects. If you have really high dispersal, it essentially makes the regional scale the local scale, and fluctuations can take you in really weird directions that you don't get rescued from. So I hope that answers that question. I'm sure that's something that we could have a discussion about later on too. So there's, it's definitely not an accepted fact that this is the only thing that happens when you crank up dispersal. Okay, so how does scaling of compositional stability differ amongst ecosystems and organisms? So this essentially is the question that our working group is focusing on right now with a meta-analysis that we're working on. And so we're looking at data from different LTER sites. And so our meta-community synthesis working group has been using a number of different methods for data discovery to find meta-community data sets. So essentially data that has community composition at the local scale we're hoping for at least a 10-year period, but we'll see what sorts of data sets we end up with. For a number of localities that can conceivably be put together into a meta-community. So data sets that make sense for scaling local stability and composition to something that helps us understand what's happening with the regional species pool in that data set. And I'll give you some examples of what these data sets are like in a minute. And what we're interested in doing is comparing meta-communities from across different ecosystems that are represented in the LTER and also across different organisms that represent meta-communities with different characteristics, or potentially end members that you might not want to consider a meta-community, but kind of give us a good outlier in terms of how our metric for community turnover would perform, like if you crank up diversity a ton and try and calculate the metric for bird data or something like that. So we're interested to address the question, do species dispersal characteristics predict biodiversity stability, but also the scaling metric for community turnover? And does environmental variability predict biodiversity stability? So this is actually something that at the end of my talk, I'd be interested in hearing ideas from folks about, because we've been trying to come up with a good way to characterize environmental variation across our data sets as sort of like a potential underlying covariate or potential explanatory variable that would explain meta-community stability. What I'll show today is that we focused in on temperature, or we took standard deviation in temperature because it's the variable that we could get a time series for from all the sites. But yeah, we're still kind of grappling with how to deal with this, and input from folks on the call today would be welcome. So the data sets that we currently have some results for include Florida coastal Everglades, fishes, diatoms, and soft algae. For Hornada, we have reptile data. For the McMurdo Dry Valleys, we have diatoms and streams. For North Temperate Lakes, we have fish and zooplankton, and I don't think we use the macroinvertebrate data for this analysis. For Santa Barbara Coastal Reserve, we have a lot of different data sets, and it includes fishes, invertebrates, and algae. And in Iowa Ridge, we have our herbaceous plant data set. And our goal for our upcoming workshop is to get more data sets that represent sort of each of these functional groups, so primary producers, consumers, predators, that live in similar ecosystems. So we want to try and get multiple data points representing kind of each trophic guild and each ecosystem. But for the time being, this is sort of where we're at with our analysis. And so today I'll kind of walk you through how we analyze the data set, and we'll focus on Santa Barbara Coastal Reserve data. So SBC, the Santa Barbara Coastal Reserve LTER, is located right by NCs. And I don't work at this LTER, so if Tomas or Max or anyone is on the call, they can correct me if I'm wrong about any of my description here. But they focus on sites that have giant kelp forests and the communities associated with giant kelp forests. And also, interestingly, there's dynamics with patches that have giant kelp forests and then they go away, and you have a totally different community, and then they conceivably could come back. So it's a really interesting dynamic system. And in these systems, there's the data that we're using have biodiversity for invertebrate grazers, many of which are passive dispersers. We also have data for fishes. So this is a predatory trophic guild, and many of these are active dispersers. And so for these data sets, they look somewhat like this, where we have total community biomass at each site. So each one of these panels represents a different site at the LTER. And each of the colors represents a different species. And then we have data from 2004 through 2016. So this is a time series of changes in the different biomasses of different species at different sites. So this is an ideal data set to address the sorts of questions we're interested in. So that's why we're using it as our example data set here. And so we can take the change in species composition through time for each of those sites. And here we've plotted them in an NMDS ordination, just so you can visualize how, how community composition at a patch changes through time. So each color represents a patch, and its trajectory through species space represents turnover and community composition at that patch through time. And so we can, for each of these patches, we can describe or come up with a way to calculate alpha variability, so the patch scale variability, by quantifying this variation through time. Additionally, we can take the data from each of these patches and come up with some estimate of what's happening with biodiversity, or with biomass and biodiversity at the metacommunity scale. So some estimate of the gamma scale variability in total biomass. So this is variation in total biomass through time. But then we also see fluctuations in species composition within the regional species pool. So this is our gamma scale estimate of metacommunity stability. And so we can add compositional turnover at the metacommunity scale. That's this bolded arrow here. And so from that, if we characterize variability through time, we have our gamma variability metric. And then after we've calculated gamma variability and can calculate mean alpha variability, we are proposing that there's a similar scaling relationship where we can divide gamma variability by alpha variability and get some scaling factor. So a phi that's analogous to what Wang and Leroux have described for an aggregate variability. Okay, and so this is the work that Tomas has really led with our working group. And he's derived two different types of beta diversity based metrics for calculating community variability. So the first, we applied Hellinger distances to look at variability at each patch. So that's following the Legendre and de Cazares method. And so essentially we can get local variability and gamma scale variability by using these distance metrics. So you can think of it as like temporal beta diversity at the regional scale and temporal beta diversity at the local scale. And by dividing those two metrics, we get our beta diversity scaling factor. However, when we do this, it doesn't really follow all the rules of a true synchrony metric. So we think it's an informative ratio, but it can take on values greater than 1, which may be hard to interpret in some cases. So there's also a method where instead of taking relative abundances at each point in time, we scale abundances to the total biomass summed through space and time. And then we get a metric that behaves more like a synchrony index. So the advantage to this metric is that it's a truer synchrony metric. However, the advantage to this metric is that you can use it with relative abundance data, whereas this metric down here, you actually need biomasses or abundances, which aren't always available in data sets in the LTER repository or other places. All right, so I have a question about, do we trust making a lot of inferences from the NMDS? And the answer is no. We don't actually use that MDS for anything other than visually representing kind of conceptually how we're thinking about diversity amongst the patches. So the NMDS was just illustrative. So really, we're using these Hellinger-based metrics instead. I have another question. Why do we not consider gamma equals alpha plus beta diversity? And it's not that we don't consider that. It's more that we're, so far we've only been deriving a multiplicative relationship between regional and local community compositional turnover. And basically we're doing this to expand upon the Wang and Leroux framework, which is multiplicative. So there's just not as far as we don't have a good idea of how we would apply the additive framework to look at composition, but we're totally open to suggestions if they're out there. Okay, so as an example, for our data set here, we calculated the aggregate biomass synchrony metric that we described earlier. So this is the same metric that Wilcox et al. Were calculating for their data sets and that Wang and Leroux derived in their paper. And so when we calculate the aggregate biomass synchrony, so this is how does variation in the total biomass at each patch, when you aggregate that to the regional scale, it's a comparison of regional variation to local variation. And so essentially you can think of this as the variation in the total meta-community is 20% of the variation that you would observe if you look at a patch or locality within the meta-community. And so similarly with our compositional ratio and our compositional synchrony metric, we get a metric of about 0.5. And so in this case, the compositional variability we observe at the meta-community scale is about 50% of what we would observe at the local scale. And these bounds here are based off of randomization tests. So you can see that the synchrony metric that we're seeing for, or the value that we're seeing for each of these metrics represents a meta-community that's more synchronous than you would otherwise expect if things were just varying randomly. Now, so this is just kind of an example of how we're applying this metric or how we can calculate it for a data set. So we're not real sure yet how to interpret this. Like, we have no idea if this is an outlier or not. Like, if this means that the system is more synchronous than your average ecosystem or less, because we've only really calculated it for a handful of data sets. But you know, we're presenting our metric here just to show you sort of what we're doing with this working group. How do we randomize these data sets across time? It's a good question. So the randomizations were performed by independently shuffling the biomass of each species within each community, keeping species temporal mean and variance constant. And I'm not entirely sure, but I think, so Tomas did these randomizations, and he may have used one of the packages in Vegan to do that. But we're happy to either take recommendations from people on, you know, how to randomize the data, or talk about or further describe how we randomized it. Okay, so you know, that was how we applied our compositional turnover scaling metric to one data set. But really our question is, how does this vary across ecosystems and across organisms? So I just showed you what we did with the SBC data set. We actually had a number of different SBC data sets. And we have a handful of data sets from across the LTER that I can talk about today, but we have many more sort of in the works that we'll be adding to this analysis. So I want to emphasize right now that the results that I'll be showing you all kind of illustrate how we're planning on approaching this meta-analysis, but there's not really any conclusions to take away from the results yet, because it's really only a few data points in the meta-analysis to this point. So as I said, we were able to get data from, this is a data set associated with SBC, but it's at the US Virgin Islands, Florida Coastal Everglades, Granada, McMurdo, MCR, so another marine data set, North Temperate Lakes, plants from Niwot Ridge, and then the data from Santa Barbara Coastal Reserve. So each of these colors is a different LTER site. And of course, there are a whole bunch of different ways to slice this up. Like we could look at functional guilds, we could look at biomes. So that's sort of one of the objectives for our coming up working group meeting, is how we'll approach this meta-analysis. For the time being, I'm plotting in this figure marine sites versus terrestrial. Terrestrial meaning terrestrial and freshwater. And so we have gamma variability calculated for each site. And I want to point out here that I used the Hellinger version, not the kind of true synchrony version, because a lot of these data sets had relative abundances and not total biomass, so we couldn't calculate the truer synchrony metric. So we have gamma variability for all these different data sets. We also calculated mean variability for all these different data sets. And then from these numbers, we can then estimate our, what we're calling our synchrony metric, but in this case, it's not a true synchrony metric. So it gives us some idea of how local stability scales to regional stability, or in this case, local variability affects regional variability. So if we're looking at the y-axis along this graph, that's essentially asynchrony to synchrony. And then along the x-axis, we're looking, in this case, low temperature variability to high temperature variability. And you know, at this point, we're not really sure what to make of this data set. So unsurprisingly, marine data sets have much lower variability in sea surface temperatures than terrestrial sites have in air temperatures. So this was one way for us to, you know, organize the data preliminarily. Interestingly, there's kind of this, you know, potential pattern or relationship between variability and synchrony and community composition in our terrestrial sites, but I have no idea if it'll end up being statistically significant or not once we have more data. So like we were saying, this is only a handful of data sets, very preliminary. Another way to look at the data is we can take our data sets and divide them up into dispersal guilds. So again, clearly there's not a statistically significant difference between these lines. But the interesting thing is they go in the directions that we expect. So you can think of this as sort of our hypothesis is that active dispersers may show this relationship and passive dispersers may show this relationship. And we're not really sure what we'll find when we have a full data set to analyze. Let's see. A quick question is that about our x-axis here. So it's standard deviation in, okay, yeah, about our standard deviation in temperature. So for the terrestrial and freshwater sites, we took air temperature data, I believe, for all of them. So it's more of just what's the standard deviation and kind of climate at that site. For the marine sites, it's standard deviation in sea surface temperature, and it's the standard deviation of the annual means. So it's a very coarse metric for variability in temperature. And again, this is one of the things where we're totally open to input on kind of how we can improve what we're using on this x-axis. Because right now for, when we plot it as marine versus freshwater and terrestrial, I don't know if it's really meaningful. Of course, sea surface temperature varies less than air temperature on land. So it kind of just splits up the biomes. Okay, so back to this figure, we have, our hypothesis is that active dispersers will show increased synchrony with increased environmental variability. And that's because if you're an active disperser, you choose where you go. And so if the landscape is showing more variability in space and in time, you're going to have active dispersers seeking refuge in the same places at the same times. So that's kind of the mechanism by which we expect synchrony there. And this will make the species pool less stable. Alternatively, with passive dispersers, when you have a more variable environment, that dispersal rate is lower. You can have refuge communities in different places and more patch dynamics occurring, more rescue effects occurring, more of sort of a portfolio effect occurring. And so we would expect an opposite direction in this relationship between synchrony and environmental variability. So that's sort of our hypothesis. And right now there's, we can't say anything from the data, but it'll be cool to see how this graph changes as we add data points to it. Okay, so back to kind of putting this all in context. So we have this approach that we're taking, looking at scaling of compositional turnover in metacommunities and how it could provide a tool that we can use to apply the metacommunity concept to assess biodiversity kind of in a macro system context. So what I mean by that is we can take this analysis approach that we're taking, and it gives us information about cross-scale emergence. So we can compare across LTER sites how the strength of cross-scale emergence varies or what the nature of that cross-scale emergence is. And then we're also interested in ways to think about these cross-scale interactions. So how does regional climate variability affect the sorts of things we see at a metacommunity and these dynamics that we observe? So this is sort of where we fit in the macro scale context and where we're going with kind of the questions we're asking with metacommunity data sets. Okay, so kind of before we open up to questions, I want to give a shout out to some sort of adjacent projects that have been super helpful to us. So Tom Miller and Aldo Camponi have put together this Poplar R package. And none of the data sets I showed today were from this package, but we have a huge number of potential data sets that we found using this package. So what it is it's a database of population data sets from the LTER that they've put together that's searchable. and in a standard format. And of course, a lot of the population data sets have multiple species, which make them amenable to a community analysis or meta-community type of analysis. So I want to direct you all to the popular GitHub page if you're interested in it, or contact us and we can put you in contact with that group if you're interested in their how to use their R package. So it's been really helpful. Also, there's the EcoCom DP project that's being led by the Environmental Data Initiative, EDI. And so they're the group that manages the repository that LTER data are in. They've put together a standard for community data that you can find at this EcoCom DP GitHub page. And this is under development, but what they're doing is they're putting together an R package that can help you format a data set to fit the data standard. They're working to have functions that can pull raw data from the EDI repository and put it into the standard data format, and then re-archive it as kind of a new processed data set. And also they're working on functionality to search and pull in data from the repository that's in the standard format so that you can use it in an R session. So essentially it's this really cool archiving and data discovery tool. So yeah, these are two tools that, you know, for anyone interested in synthesis science, I think are really handy. And then we have a couple of future meetings. So our next working group meeting actually is next week. So a lot of this stuff that I've talked about today, hopefully we'll be making, you know, more progress on, and those figures will look totally different by the end of next week. And then we want to put together our last working group meeting to focus on kind of more forecasting meta-community stability and maybe doing some modeling. And so that's something that is sort of still in the works. But if this is something that folks are interested in, please email me and I'm happy to kind of talk about it, or if you have ideas of directions we could go with it, this is still being planned. So I'm happy to hear feedback on it. All right, so I'd like to acknowledge the LTER network and NCs and the NSF for providing support for our working group. And I'll take questions.
Thank you so much, Eric. And while Eric is answering questions, you're going to see a small poll. Those of you who are still on, if you can just stay on another minute and let us know how you heard about the webinar series, that would be great. The poll will go away if you answer the questions off your monitor. And Eric, you do have a couple questions piled up in the Q&A bar, including one from about the middle of the webinar from Gabrielle Sousa.
Yeah, so yeah, that first one, let's see, may I use one estuary as a regional scale and estuarine zones as a local scale? So really, you know, it kind of depends on your knowledge of the dynamics in that system. I mean, I'm not a marine scientist, so I don't know. And that may be apparent from my description of the marine data set that I was just talking about. So, you know, if you have a hypothesis that makes sense at those scales, then yeah, that's totally appropriate. And actually, I think it'd be, so the new Chase and Leibold book, or Leibold and Chase book, I forget who's the first author, on metacommunities, they talk about this idea of sort of different, the continuum of discrete versus continuous metacommunities, and also the idea of like what scale are you applying the concept. So, you know, this is something that there's no hard and fast rule about, and I definitely suggest taking a look at, you know, their book, and they have some good insights about these questions. But generally, I think it's a good framework for testing hypotheses. So if you can, you know, come up with a good hypothesis about why you would want to classify it that way, then it would definitely be appropriate. Let's see, how do I discriminate patches in a marine system? That I have no idea. So I basically deferred to Tomas Lamy, because he's a marine scientist and I'm not. And I don't know if he's on or if Max is on. If one of you want to answer that question, feel free.
Eric, I don't see Tomas or Max. I did see Andrew Rassweiler, but I think he's gone now.
Okay. Yeah. So yeah, that's one of those things where I'm not familiar with that ecosystem. I just defer to the experts on it. But if you want to email me your question, I can forward it to them and get your response. Let's see. Environmental variability. Okay, so there's a question about environmental variability. Does it act as a filter? And then synchronizing the community. Okay, I think the question is sort of what's the mechanism for linking environmental variability to metacommunity variability. And so, yeah, there's sort of a couple of mechanisms by which that could occur. So that's something, I guess that's something I didn't really talk about too much in depth. So there's kind of a number of questions we could address. So one is, what sort of metacommunity paradigm occurs in a given ecosystem? So that could be related to how variable the environment is. But then there's also kind of the direct mechanistic link between the environmental variable and what's occurring with community assembly, which would be, so that would directly be the species sorting paradigm. And, you know, in some ways they're sort of confounded, because if environmental variability affects the relative importance of species sorting, but environmental variability also determines species sorting, there's not really a good way through an observational study to tease that apart, as far as I know. So on the other hand, though, like in a meta-analysis framework, we're hoping to see like some patterns emerge where maybe one sort of pattern persists more at one end of the environmental variability scale, and another sort of pattern persists more at the other. So that's something that it's definitely a great question for fodder, or a great question for discussion at our working group next week. But I don't have a good, I don't have a good explanation in terms of like what we think that mechanism is. Let's see, so with this work in fire event prone driven systems, return frequency at 2 to 5 years. So, you know, that's a good question. And actually one of the things we want to do moving forward with our working group is also find data sets where discrete disturbances are identified, and we can sort of categorize them in terms of like the return interval of disturbance. So it's something that we're interested in approaching, or a question that we're interested in addressing. And so that actually reminds me that I wanted to kind of solicit folks for input. So for each of the LTER sites, we're trying to get input from people that know the system, so that they can identify, you know, or essentially give us their local expertise and tell us if disturbance recurs, you know, at a known interval, what were the known major disturbances. So for example, at FCE, LTER, I know there was a cold snap that had a dramatic influence on a lot of the aquatic species not too long ago, like in 2010, I think. And that Hurricane Andrew was a big disturbance event. And so that's the sort of thing where for each of these data sets, we think knowing that background is important. So, so if anyone has like intimate knowledge and is like interested in sharing that with us, we definitely want to know, want to be able to get feedback from folks. And I want to give a shout out to Luca at the FCE LTER. He also said Hurricane Wilma had a big impact on LTER data. So see, this is a good example of like getting someone who is an expert at the system, getting their input can give you an idea of the important disturbances at a site. Also, if folks have leads on data sets they think that would be appropriate and good for adding to our meta-analysis, or if they have ideas that they think we should consider, again, feel free to shoot me an email.
Great. Well, thank you so much, Eric. And I'm sure the audience is giving you a big virtual round of applause for such an interesting, thought-provoking seminar. And I'd just like to remind those of you who are still on that these, if you'd like to watch it again, or your colleagues might be interested, these are recorded and you can find them on the LTER NCO website. Oh, there's another question. So before I cut you off, why don't you take that last question, and then after this one, I'll encourage folks to email Eric directly.
Yeah, this is a great one. So what is the temporal scale of our data? And so for this, for these plots that I showed in this talk, we did annual scale, but it's something that's been a debate in the group for sure. And, you know, we're kind of going with annual for this meta-analysis, and accounting for things like, well, what we'll end up doing is accounting for like life cycles or species, the length of species lives as kind of a covariate, because, you know, clearly, you know, semi-voltine things are going to respond much differently over time than univoltine things, than multi-voltine things. So, you know, if you have many births throughout a year versus one birth every 5 years, you know, it's going to be really different. But yeah, that's one of the challenges in the synthesis project, is dealing with the different temporal resolutions. So yeah, it was annual, but there's definitely an argument for doing different sorts of timescales.
Great, and great questions. And again, thanks to everybody for attending, and thanks to Eric for presenting. Our next in the series will be Lauren Hallett and her group on June 7th. So you can find information on our website, register, and same time, same place, Zoom, June 7th. So thanks again, Eric.
Yeah, and thanks everyone for tuning in.